Autophagy from the Yetila group with the title A Comprehensive siRNA Screen for Kinases that Suppress Macroautophagy in Optimal Growth Conditions. So first of all, I would like to give you an introduction, move on with the screening strategy, then tell something about the validation of the candidates and end up with the summary. So the introduction. The whole basis of the study is that, as you all know, autophagy is a really complex process which removes misfolded proteins and organelles from the cell, but so far relatively less is known about the exact regulation, so that's why the group wanted to know which processes are involved in this important process. So the screening strategy, they basically used human MCF7 breast carcinoma cells and transfected them with kinases, also with human kinases, and then they counted the number of LC3 punctae which correspond to an increase in autophagy. So that means the final outcome are then negative regulators of constitutive autophagy or positive regulators of autophagosome maturation. And now you might ask, how does this look like in reality? And here you can see what they get. So they finally could identify 10 kinases as autophagy regulators, and you can clearly see that on the left-hand side, on the control, there is no or almost no positive punctae corresponding to an increase in autophagy, whereas on the right-hand side, in the upper panel, in the lower panel, raptor, that's a positive control, you can see that LC3 accumulates. So then the validation of the candidates, they basically used one important method, which is a dynamic method, to measure the autophagic flux. And here they used the tandem construct, which consists of mRFP, eGFP, and LC3. And with this construct, it's possible to make early autophagosomes visible, which appear as yellow, so it's a combination of RFP and GFP, and late degradative autophagic vacuoles appear only in red because of the quenching of the GFP signal in the acidic lysosome. So here just one example. The group claims to see that there is an increase of initial autophagic vacuoles, which are shown here in green, whereas they could not see an increase of autophagic vacuoles in the late stage, which are shown here in red. So then they concluded that link 2 must inhibit the autophagolysosome maturation. Then let's sum up. I showed you today that the group performed an siRNA screen which identifies kinases that regulate autophagy, and with the use of a very powerful tool, the tandem fluorescence LC3 reporter, they could validate their candidates, and here I showed especially one example, which is link 2, and this is important for the autophagosome maturation. So thank you for your attention, and I'm happy to take questions. And now not even 5 minutes.